Okay, good morning, everyone. This is Cherry Kubota, accessing from the Ohio State University. Welcome to Indoor Ag Science Café, January. So I'd like to have John Erdo, my co-host, organizer of this cafe. So the Indoor Ag Science Cafe is funded by USDA NIFA Specialty Crop Research Initiative Program. We are getting into the fourth year. The project director, Eric Ranko, is here today also. And then we are so excited about every month having this opportunity to conversate, to have a very important conversation to move the industry forward. So anyway, like to have John, and then John, you can introduce a little bit of yourself, and then introduce the rest of the slides we have at the beginning.
All right. Well, thanks for everybody for being here this morning or afternoon, depending on where you're at. I'm John Ertl. I'm a PhD candidate in Dr. Kubota's lab. I'm just going to take you through a little bit of the introduction here today. So we are the Indoor Ag Science Cafe. In the upcoming series, we have today Harry Burman of Rijk Zwaan USA talking about introducing spinach in the hydroponic crop production. In just 2 weeks, Dr. Kubota will give a presentation herself about low pH hydroponic nutrient formulas for growing leafy greens to better resist root rot disease and manage that risk in hydroponics. On March 7th, we'll have a presentation by Dr. Selena Gomez of Purdue University about indoor gardening. And if you'd like to sign up or find out more about any of these events, you can look at the top right corner at the OptimIA events page. And so that's scra-optimia.org. And you can find a lot of our information there, as well as a repository of the previous presentations from the Cafe series.
I just wanted to inject here. So I realize my title tends to be long compared to other people. But I put that as much as information why we wanted to grow, leafy greens at low pH. So anyway, just wanted to comment. And then also the Dr. Gomez presentation title is tentative right now. I know she's gonna talk about this, but title might be updated in the near future. So that's it.
All right, thank you. Okay, so something else I'd like to draw your attention to, again, in the top right corner is the link to optimiauniversity.org. So what we tried to do here was create a knowledge base for the indoor farming sector. A lot of information about the background of indoor farming systems, plant responses to environmental conditions in indoor farms, crop-specific production management, economics. So just a well-rounded picture of a lot of the concepts to run and understand the science in successful indoor farms. And so if you'd like to learn more, there are going to be, and there are already plenty of interesting lectures and short videos to describe these various topics. So please visit that site and share with people you like in this industry, and we're happy to provide a lot of this information to you guys. All right, today's agenda. So this is going to be 2 parts. The first part will be a recorded presentation by Harry of Rijk Zwaan about spinach hydroponic crop production. We will at that point end the recording once Harry is done talking, and we're going to have a Q&A sort of session that I'll help moderate with Dr. Kubota. So that won't be recorded. So if you'd like to stay and ask Harry any questions, that's the part that's going to be important for you to hang around for. And it'll be immediately following the initial presentation. So with that, I'm going to stop sharing. And Harry, you're welcome to start sharing, and you're welcome to begin your presentation.
Okay. Yeah. Thanks, John, for the introduction. Yeah. So I'm Harry Burman. I work for Rijk Zwaan. I'm a crop specialist. So my I'd say 95% of my job is focused on hydroponic lettuce. It's mainly sales, but also trials. So getting information back to the breeders so we can improve on our varieties. But increasingly, I have more demand from spinach. I mean, I can't visit a grower without them asking what spinach do we have, what are we doing in terms of research. So there's huge demand, and we've been testing a lot of our varieties and different ways of growing it. So still a lot of progress to make, but I'm here to share with you what we have so far. And how do you make the slide? Here we go. Okay, so yeah, just as mentioned, I'll just give a brief market overview of what I see across US and Canada. The trials that we're doing in Holland, previous trials, and then we currently have one running right now as well. And then observations in the field about hydroponic production and water management. Yeah, so as mentioned, and I'm sure everyone knows, the demand for spinach is very, very high. That's mainly coming from the retailers. They're increasingly getting a larger percentage of their lettuce from indoor agriculture, and they also want the same growers to supply them the spinach. So they don't want many different growers supplying different products. They want generally a handful of suppliers that can supply them multiple different products. And then on top of that, they also have disease issues similar to what you see in the field with the lettuce. So anytime there's a recall, that's huge loss to the retailers if you have to pull all the spinach off the shelves as well. So similar story to the lettuce, but yeah, no one's doing it on a large scale yet because of the root issues, as I'm sure everyone knows. Yeah, so these we did some trials back in April 21, so quite a while ago. These are the first trials that we did with spinach, kind of preliminary test just to test our genetics. And then, as I mentioned, that we're also currently running a second round of trials, which I'll go over later in the presentation. So this trial was to really see different germination rates of our varieties. So is there a difference between the genetics as well as the treatments that we'll apply to them? So they were all grown in jiffy pots. This was in a trial station in Belgium, and under an independent contract researcher. So they conducted the trial. We tried to not influence it as much as possible. So it was as least biased as possible. We used naked seeds, 7 seeds per pot, and then once we planted them, we covered them with a light dusting of sand just to keep the humidity higher around the seed. We had 20 treatments, 65 replications, and 6 cycles. We started 4 days in the germination room, which is very crucial to getting a good start. We find that spinach really needs its own dedicated germination room. You can't just seed them out in the greenhouse. They're very sensitive. And then once they're germinated, they transfer to a plant raising table. We tried to keep temperatures below 20 C. This was in the germination room, by the way. Max 20 degrees Celsius, and then very high relative humidity above 95. And then we covered all of the trays with styrofoam to keep that humidity inside of the trays. So these are the treatments. I can't tell you the priming that we applied. There's different types of priming. We tried 2 different types in this trial. I'll just call it priming 1 and priming 2. And then we also added an organic seed to see if there was a difference as well. And then, of course, compared it to untreated. And then we tested our 6 main varieties that we have at Rijk Zwaan, and then 15 others just to try and screen more of our material. But they did not have, the 15 other varieties did not have the priming applied to them. It was just to see germination differences in the greenhouse. So in the greenhouse, tried to keep it below 20 degrees and above 16. Day length varied 12 to 18 hours. It's depending on, I'm not sure actually why that's such a wide range of lighting. I think there were some issues with the trialing station. But yeah, that's quite a wide range of day length. Yeah, in the day, relative humidity 70 to 80, and then 85 to 95 at night. And then we applied CO2 1,000 ppm in the day, and then none at night. It was 4 days in the germination room, and then 10 days on the plant raising tables at a density of 150 plants per square meter. This is low density just because we were looking also doing a transplanting trial. So we wanted to do 7 seeds per pot and then try growing them for longer periods of time and doing a transplant to increase yield instead of just doing a direct sowing, 1,000 seeds per square meter, which is quite common in the industry. So this is another layer of trialing that we did on top of that. So that's why the density is quite low. And then another 10 to 12 days in the growing system. And then the end density was about 40, it says plants, but it's really pots per square meter. So, kind of what I'd call teen leaf density, which is part of the reason why we want to see if it would fit in a teen leaf system. And then also if we could automate the harvesting on top of that as well. So germination, not as good as lettuce, but there's quite a big variation. There's mostly around 50 to 70% with a max of 87%, and this is all treatments. So yeah, as I mentioned, quite a range. Yeah, and here's the results of the germination trial. So yeah, this is on the on the bottom there's the date. So these are the sowing dates. So for example, a 30th of April was one planting date, and then the next planting date was 3rd of May, and then so on. So we did six plantings, and then recorded the germination over time. The different colors correspond to different varieties, and then the results were that, as expected, the untreated had overall lower germination rates. Surprisingly, the organic seeds had there's quite a bit of variation, but yeah, quite higher germination than we expected. The lowest germination was in the lizard variety. If no treatment was applied, that was the orange line all the way down below 20%, so not very good at all. And then we found that priming 2, the second type of priming, showed more variability over time. So it's quite closely tied together, and then over time it had more variation. So it seems like priming 1 is a bit more stable than priming 2 for germination. Yeah, these are the results. So different colors mean significant differences. So pilot fish had the highest germination rate, and then Sun Angel, and then priming 1 had the best results compared to priming 2. So yeah, these are the conclusions. What we found is different priming methods had different effects on different varieties. So one form of priming could increase the germination rate of one variety, but not another. and vice versa. Let's say the second priming technique could increase one variety more than the germination more than the other. So a lot of variation must be down to the genetics, of course, but interesting to know that not all priming is equal. So different priming methods affect different varieties differently. So yeah, we found that generally it was 20% increase in germination, and pilot fish and Sun Angel overall had the highest germination, which is good because Sun Angel is also one of the better varieties for growing commercially. So there's just something about that variety that is robust in terms of growth. And we also see that in the field. It's one of the better ones in the field for stress tolerance as well. So that's the one that we've been focusing on mostly. These are just some pictures from the trials. These are early stage. These are from the first cycle, so they look great. This is Platypus, Sun Angel, look very healthy. Unfortunately, they didn't last that long. didn't look like this the whole trial. Lizard, and then, as everyone knows, after a few cycles, you start to get these yellowing leaves. And then, yeah, essentially you get the whole system crashes. And then, as a test, we also put in some different plants. So on the left is beet leaf, in the middle is kale, on the right is arugula. Kale obviously is whatever is happening in the system in the water is extremely resistant to it. It's bright white roots, grew like there's nothing going on, whereas the arugula was very affected, and then the beet leaf was somewhere in the middle. Yeah, so that was just one of our first trials, just to see germination rates. We have, as a seed company, we have different product forms, different treatments we can do on our seed. So we're looking at not just priming, but there's various other applications that we're doing on Sun Angel to make it more robust than just a standard priming. So we're making a hydroponic product form which has multiple treatments added to it to make it more harvest ready. And then on top of that, we're also looking at what's going on in the water. So the trials that we're doing in Belgium right now are more focused on filtration, testing what's going on with the root decay, and seeing if there's ways to mitigate that. Unfortunately, the trials are about a year behind. It's been a headache trying to organize everyone. There's multiple stakeholders involved, and the trial has only just started. So we're hoping to get more results by summer through various different trials that we're doing around there. So hopefully in a year's time, or at least 6 months' time, we'll have more focus and more knowledge about what's going on with the root decay. And then, yeah, as this slide mentions, this is really the focus of what we're doing with hydroponic spinach. We need more harvest sure varieties, higher germination rates. You know, even if priming does improve germination rates, I think we can still get them higher than they are. That's you know, when lettuce is at 95%, you know, it's I think we should aim for those levels, really, because it's, you know, it's a waste of seed for the grower. It's a waste of labor. If you you know, you have to overplant, then, you know, it's just better all around quality, uniformity as well. If all of the seeds are germinating at the same time, if you're automating that harvesting, you know, you're getting those plants all at the same level. And if you don't want your harvester cutting small leaves in half because it doesn't look great in the packaging, and then you're getting shelf life issues, and then long cotyledons as well. You know, it's you don't want half a cotyledons in your bag as well. And then of course, stronger root system. That's what everyone is struggling with. Yellowing leaves, of course, higher yields, thicker leaves, better uniformity, as I mentioned, for mechanical harvesting. So this is more about observations in the field, and then at the end I have a bit of hypothesis as well of what we think is going on and what we're targeting. But this is just over, global. This is not just a North American issue. This demand for hydroponic spinach is for some reason it's taking off around the world. There's companies in Asia and Europe that they're also focused on this. Demand is very high. So yeah, this is a combination of observations from around the world. Yeah, I'm sure most of you know there's really two ways that spinach is currently being grown: deep water culture on floats, high density, and then on NFT in pots, as I showed in the trials. We're doing small number of seeds per pot, and then generally doing a transplanting, similar to what you would with lettuce. These are the main growing mediums we see. Elapot, great for lettuce, cost effective. You can make up your own blends. Doesn't really have an effect. None of these growing mediums seem to really help with the root decay. They all have issues, but this is just a general overview of what everyone has tried. Plastic pots. That's more Russia. They use a lot of plastic pots over there. Jiffy pots. Some growers have tried that in North America. Rafts, that's probably the most common that I see. High density, direct sowing rafts, and then rock wool is not recommended at all. It's not the right medium. It holds too much water, and spinach does not like wet feet. So what we see is similar to lettuce is if you can control the climate and make the climate softer, easier for the spinach, the better. So I kind of say that lettuce has a certain growing band, and then spinach within side of that has a smaller growing band. And so there's a very fine line that you can grow in with spinach, it seems. If any of the variables, it's too hot, water temperature is too hot, or it's too humid, air temperature is too hot, then you seem to have, it affects the spinach more or quicker than the lettuce does. So various ways to mitigate that. There's greenhouse covers, artificial light, screens, fogging, germination room, which I mentioned earlier. It's very important to have a dedicated germination room, disinfection, water treatment, and then heating and cooling. That's both of the air and the water. So yeah, not a short list of equipment needed. One thing that I think is very important, goes for lettuce as well, is diffuse greenhouse cover. You generally see improvement all around. You get thicker leaves, lower tip burn for the lettuce, of course, less stress, more uniform production. It's just a bit of a softer climate inside the greenhouse compared to standard greenhouse covers. This lighting, this comes from lettuce, but I think there's some research needed for the right light levels for spinach. I mean, every greenhouse I visit, it's just the spinach is in the lettuce greenhouse, and of course you're going to prioritize the lettuce growing because it's the largest percentage of the greenhouse. But I think there's some improvements that can be made, different light spectrums, quantities. I see if there's too much LED, especially I see this in vertical farms actually, if it's 100% LED, spinach tends to cup, and I've not heard of a good theory of why that is. Maybe it's too high light levels compared to temperature, and it's kind of it's over producing sugars. I'm not sure, but I think there's definitely some room for improvement. And then screens, energy screens. If you want to keep the temperature in your greenhouse, shading, these are really good for transition periods. So you know, if you have really sunny periods and you know you're coming into cloudy weather, that you can start pulling the shade a bit more to help the plants transition into that shading, that shady period, and then blackout if you want to light your greenhouse while it's without annoying your neighbors, essentially. Yeah, fogging. See this more and more, especially in hot climates, especially in the south, where it's difficult to cool the greenhouse. In summer, when it's really hot. Yeah, the issue is that spinach cannot get too wet. It does not like too wet feet. So you have to be very careful when using a fogging system, a misting system. But a great way to control the temperature and the plant temperature in the greenhouse. This is extremely important for spinach, probably one of the most important. I've never seen spinach, maybe someone else can correct me, but I've never seen spinach be able to be grown above 22 degrees Celsius. It seems to be very susceptible to oxygen levels in the water. So, you know, as temperature goes up, oxygen goes down, and unless you have, you know, an injection Moliere system, for example, you know, it really has to be below that 22 degrees. And then in winter, above 18, otherwise the growth rate slows down significantly. So that's yeah, it's not a large bandwidth, but yeah, that's what we see. Germination room, yeah, as I mentioned, very vital for a good uniform germination and emergence. High humidity, and depending on the product form, you know, if it's primed as mentioned, maybe you can shave a day off. Generally, it's around 4 days. Maybe you can shave a day off with priming down to 3, and then with, you know, if it's not optimal, maybe you get up to 5 days in the germination room, and that is generally between 16 to 20 degrees Celsius. So here's where we get into a bit of hypothesis. That, of course, everyone is focused on Pythium, but we think that's a secondary infection, that there's something going on with root exudates with spinach. And yeah, here's the slide. Yeah, so when the plant is stressed, it produces more root exudates. And as I mentioned, spinach seems to be very susceptible to stress, and whether that's environmental, oxygen, especially oxygen in the water. If there's any fluctuation, it seems to be able to produce or produces more root exudates than lettuce, for example. And it seems to be more sensitive to it. So, you know, spinach can show stress when, you know, a lettuce plant right next to it can show none. So it does seem to be more sensitive. And then we believe we have done some preliminary water samples, and we believe they are of these fatty acids. As some of these come from the plants, from the roots of the spinach. And then, as everyone knows, you know, when you have a brand new system, you can do a few cycles of spinach and it looks amazing. But the more cycles that you do, you know, eventually you'll crash. It seems to be there's this like 3 to 6 month window where you can grow spinach, and then something builds up in the system, and then everything crashes. And we believe it's these fatty acids that are building up in the water, and they get to a toxic level for the roots, and then the roots die off. creates more stress, and then you go into a downward spiral. So yeah, I've covered a lot of this in the presentation already, but to mitigate these root exudates or the stress of the plant, that you need to be able to create enough oxygen, especially in the night, because the oxygen levels during the night can drop off rapidly, especially if you have algae in the system. They really absorb a lot of the oxygen, the dissolved oxygen in the water, and then you can, so you can get good oxygen levels in the day, but not realizing that your night levels are below optimal, or at least damaging your roots. A good irrigation strategy, it is best to have spinach on its own. This is, I guess, more for NFT. Its own irrigation strategy, not one that's on the same one as lettuce, because lettuce can take more water, more irrigation than spinach can. They like to be a bit drier. So if you can control the irrigation on the spinach, that is much better. And then, yeah, I mentioned good algae, keeping algae levels low. Still water, that seems to be an issue that ties into oxygen levels, and then biofilm as well, controlling biofilm. Yeah, and these are just the ways that we're looking at controlling these fatty acids. This is actually currently what we're doing in Europe right now. We're looking at active carbon. There has been a grower in Belgium that has used active carbon before, and is seeing great results. The problem is that the carbon gets saturated very quickly, and you have to keep replacing the active carbon, so it gets very expensive. So it seems to be that there is a solution right now, but it's just too expensive to do. So if you have to replace your active carbon every month, does it make sense? Can you make money essentially with by growing spinach? And then beneficials, I think this is very important. So the question I ask myself is, why can spinach be grown in soil multiple times? I think 2 or 3 times back to back, and it'd be fine. So I think there's something going on in the soil that may be dissolving these fatty acids or mitigating. Maybe there's some bacteria that is biodegrading the root exudates. I'm not sure. This is just all theory, and so maybe we can kind of replicate some of that in the water. Maybe we can build up some ecosystem bacteria, fungi that can really help support the ecosystem in the water. Creating water movement. There are some volatile fatty acids, so this would help with that, but there are ones that do not dissolve through water movement, but water movement does help. Sometimes we'll see if a grower's, I've seen this in Europe, if a grower's growing spinach and they create water movement in their return tank, that sometimes it starts foaming like there's soap in the system, which also is another kind of indication that maybe it is a fatty acid that is in the system that's affecting the roots. So I wouldn't recommend just one of these solutions or one of these treatments. I think when growing spinach, you need multiple, because if one fails, you're going to run into issues very quickly. So it's kind of like you need insurance. You need a combination of all of these things, I think, to be able to grow spinach successfully. And maybe, there are multiple different ways to filter water as well. We're looking at different ways apart from active carbon. That is a much cheaper way of filtering the water. But yeah, I think a combination is going to be the way to go. What combination it is and what types of bacteria, all of these details are what we're looking into to try and figure out this growing solution. And that's it.
All right, Harry, thank you so much for the presentation here.